Hi, my name is Raché Manning. I am one of Dr. Roland K's students at NC State, and I'll be discussing the possible relationships between productivity, abundance, and diversity, specifically for herbivores. So first off, to get a bigger picture of what my groupmates and I were studying this semester, I have two pictures on the next slide of habitats, and I would like you to take the briefest of moments to choose which of the two you would expect to find a higher number and diversity of herbivores. So at the top of your head, would you choose one or two? Now, of course, I'm assuming you chose picture number two, considering it has a lot more plant life than the habitat pictured in number one. So one would automatically assume that more plants means more herbivores because it just makes sense. This is more or less the basic concept of the more individuals hypothesis, which states that the more energy that is available in a system, the more individuals you can have sustained by that system, which in turn makes it possible for there to be a more variety amongst those individuals simply because there are more of them. And this energy availability is referring to plants and plant productivity. The dynamic habitat indices are aspects of productivity that can be measured and used to indicate certain things about a system. The cumulative index in particular can be used to measure energy availability in a system. So what's unknown is whether or not the more individuals hypothesis actually applies in nature, specifically for herbivorous mammals. Though logic may say so, there is not enough empirical evidence to support the theory that productivity and abundance cause diversity. So our objective was to find correlations between plant productivity, herbivore abundance, and species diversity among herbivorous mammals. We predicted that we would find positive correlations between all three, specifically with the cumulative index for productivity. We used Snapshot USA data from 2019 and treated every subproject like a separate community. We filtered out all of the camera detections that were of herbivores, which we defined as mammals that have a diet of at least 95% plant material. For plant productivity, we use the gross primary production, DHIs, or dynamic habitat indices, from the dataset. We used detection rates to measure abundance and the total number of species detected in each community for raw species richness. We then calculated the Shannon diversity index for each community to measure species richness and evenness. Now, almost all of our correlations were statistically insignificant, all except for these two, the cumulative GPP index versus the raw species richness, and the minimum index versus the raw species richness. Though these two correlations don't have the best fit linearly, they are interesting because they show slightly negative correlations, which is the opposite of what is expected. One would assume that higher species richness would be in areas where the minimum plant productivity is quite high, and the cumulative or the energy availability is also quite high. However, these results don't show that. So in conclusion, with the data and methods that we used, we could neither support nor reject our hypothesis that plant productivity and herbivore abundance had positive correlations with species diversity. We also could not support nor reject the more individuals hypothesis, which was used to inform our own hypothesis. If this were to be revisited, I would think that having a more accurate way to measure animal abundance could possibly give better results, since detection rate per subproject does not seem to be a very good representation of abundance. I also would explore the different diversity indices that exist and look more into which ones work well for certain sample sizes and data collection methods, seeing as the Shannon Diversity Index did not yield any significant correlations in our data and our results. And I hope you enjoyed the talk.